Good afternoon. Welcome back. Today we're going to talk about how to build virus particles, structure of viruses. So let's first talk about what the outside parts of a virus does. What are the functions? Main function, to protect the genome. You have to build a nice shell so that the virus can go from host to host in harsh environments and be protected. So stable protein shell. That the protein shell has to recognize the nucleic acid, so it has to specifically put virus nucleic acid in it, not cellular. That would be a waste, right? And we'll talk about how that happens. And then for viruses that have a membrane, the proteins have to figure out how to interact with the membrane, so the membrane is there. Then the virus particle has to deliver the genome to the right place in the cell. It has to first bind virus receptors on the cell surface, a topic we'll talk about next time. It has to uncoat the genome. So uncoating is a word we use to mean get the genome out of the virus particle. We call that uncoating. For viruses with membranes, the membrane has to fuse with the cell membrane somewhere in the cell. Again, we'll talk about that next time. And then the genome has to go to the right place. It's not the same for all viruses. Depending on what the nucleic acid is, some viruses, the genome goes in the cytoplasm, sometimes it goes into the nucleus, and we'll talk about that as well. So let's define terms before we go any farther. There are a lot of them today. First, the subunit. When I say a protein subunit, when I'm talking about virus particles, I mean a single polypeptide chain. And that subunit makes up the next level, which is the structural unit. And this is the unit from which we build capsids. It can be one or more subunits, or in other words, one or more proteins can constitute a structural unit. So you can see on this virus particle on the upper right, the structural unit is outlined in black, and it has a green, a red, and a blue protein. In other words, a green, a red, or a blue subunit. So that structural unit there is made up of 3 subunits, and they're different proteins. Then this is a capsid on the upper right. So many viruses are built and have capsids like this. We'll explore how they're built today, but capsid is from the Latin meaning box. It's the protein shell that's around the genome. And many viruses are just like that. They're naked capsids with nucleic acids in between. And then some viruses have an envelope or a membrane around the capsid. Sometimes it's around a capsid. Sometimes it's just around nucleic acids. So here is an example of a virus on the lower right, where we have a capsid, a protein shell surrounded by a membrane that happens to be a herpes virus. And then on the left is an example of a virus where we have a membrane surrounding the genome. The genome is not in a capsid, but it's in what we call nucleocapsids, or in other words, protein attached to the nucleic acid. This happens to be influenza virus. There are 8 RNAs in the genome, and each of the RNAs is bound to proteins. So we call those nucleocapsids. It's the nucleic acid protein assembly within a particle. We use this word, this is very confusing. Every year it confuses a lot of you, probably because I don't explain it well. The nucleocapsid is called such when it's a substructure. Don't ask me why this is the case. You know, people make up terms for stuff. They don't ask me because it's many years ago. This RNA protein assembly in the influenza virus is a nucleocapsid because it's within an envelope, so it's a substructure. Here on the right, this herpes virus, this capsid in the DNA is a nucleocapsid because it's within a membrane, so it's a substructure within the membrane. On top, the naked virus has got nucleic acid in, but we don't call it a nucleocapsid because it's not a substructure. If you wrapped a membrane around it, then that would be a nucleocapsid, all right? So just to make clear, the nucleocapsid is only called that when it's a substructure, and we'll see lots of examples of this today. Now, in terms of size, I'm going to use terms that I'm sure you have heard, but just to make sure we're all thinking the same, we will use nanometer to express sizes. A nanometer, of course, is 10 to the negative 9 meters. or 10 angstroms, or 0.001 microns. So I will use all of those kinds of sizes. Just to give you some examples, here on the upper right is the protein alpha helix. That is about a nanometer in diameter. A DNA strand, a double-stranded DNA is about 2 nanometers in diameter. Ribosomes, 20 nanometers. Poliovirus, shown here, about 30 nanometers. And Parvovirus, one of the really big giant viruses, is about 1,000 nanometers. All right, so a nanometer, 10 angstroms, 0.001 microns. Virus particles are what we call metastable. It means that they're really stable at some point to protect the genome. So if you swallow a virus like poliovirus, it's passing through very harsh conditions in your stomach and intestine, and it remains infectious. So it's highly stable, yet it has to give up the genome. At some point, it has to become unstable, and so the combination of the two is what we simply call metastability. So here we have poliovirus. The virion is extremely stable. When it attaches to its cell receptor, that makes it unstable. So there's some trigger in the reproduction cycle of a virus that makes it unstable. And we'll talk about some of those next time. But this metastability is a really important concept to understand. And another way of looking at it is that the virus particle has not yet attained its minimum free energy conformation. And it has to get there in order to come apart. And in order to get to this minimum free energy conformation, which is shown on this graph here, here we have free energy G on the y-axis with different times in virus entry into cells. And we have to achieve this point here towards the lower right where the virus particle comes apart. That's the minimum free energy conformation. To get there, there's an energy barrier that has to be surmounted. So the virus exists in a stable conformation, number one. To get to 3, it has to go over this energy barrier. And the barrier is there so that virus particles don't just come apart on their own when they're floating around in the extracellular environment. Something has to trigger that to get to state number 3. And we call virus particles spring-loaded because when they're assembled, they have energy put into them in the form of bonds. And then when that energy is then used to disassemble the particle upon the right signal. So in the case of poliovirus, it's a receptor binding to the virus particle that signals the particle to come apart and energy is liberated. Okay, so minimum free energy conformation and spring-loaded are important concepts. And spring-loading, think of a spring, right? We compress the spring, and then when we let go, it expands and gives off energy. Same thing with a virus particle. So how do we get this metastability? How do we have a virus particle that's really stable on the one hand, yet can come apart? And there are a number of components to understanding this. The first is that to build a stable structure, which is part of the metastability, we take many identical proteins and repeat them over and over to build the virus particle. I had some viruses for you ready to bring here, and I forgot them all. I wanted to show you one, but I have some pictures of them later. Next time I'll bring them. So you repeat when you repeat one or a few proteins many times, those proteins can make maximal contact with each other because they have evolved to be able to do that when you repeat them over and over, as opposed to have thousands of different proteins all together. So that gives you a stable structure. So how do we make it unstable? First, those proteins are not covalently bonded together, right? If they were, it would be too stable and it would never come apart. That's a really straightforward concept. So when they're not covalently bound, they can easily be taken apart or loosened on infection. They don't have to actually all come apart, all the proteins. They just need to be loosened, sometimes to make a pore where the genome can come through, and that will be enough to get uncoding. Right? So that's stability by symmetrical arrangement of proteins, and instability, that symmetrical arrangement is not covalently bonded, so it can come apart. That's metastability. So let's see if I explain that properly on our first question. How is metastability? Viral capsids are metastable because they must protect the genome when the particle is outside of the cell. They must come apart and release the genome into a cell. They have not obtained a minimum free energy conformation. They are spring-loaded, all of the above. Most of you got all of the above, which is correct. All these answers are right. Viruses are metastable because they have to protect the genome when it's outside of a cell. They have to come apart, right? Those are the two parts of the metastability. They've not obtained a minimum free energy conformation. We have to get there in order to uncoat, and they're spring-loaded. That's all part of being metastable. Now let's talk about the tools that we use to solve virus structures. Very briefly, just to give you an oversight, we're going to talk about virus structures today, and I want you to understand in a way how we get them. And the tools include electron microscopy, Xray crystallography, cryo-EM or cryoelectron tomography, and nuclear magnetic resonance spectroscopy. I'm not going to go in much detail on any of these, but if you're interested and you have the textbook, that's chapter 4. So that is a picture of me up at Cornell in Ithaca. A couple of years ago, I visited, and they have a cryo-EM device. That's an electron microscope there, which is fitted to take pictures of viruses at very low temperatures, because as you'll see, the whole thing about cryo-EM is that you freeze the virus particles in order to give them contrast. So this thing has liquid nitrogen flowing through it, for example, to keep it cold. All right, let's explore some of these. So electron microscopy, of course, was first used, as I told you in the beginning, in 1940. The first electron microscope invented by Helmut Ruska in the 30s, and then he used them to take the first pictures of virus particles. That's a photograph of E. coli. E. coli is the big mass on the bottom with bacteriophages attached to it. There's the paper, in German, of course, and that showed that viruses were particulate. They weren't liquids. This was a really important experiment. So you can start to get an idea of what the particles look like, and you can get low resolution images. You don't get a lot of structural detail. You can't see atoms like we will be able to see with X-ray crystallography and cryo-EM, but you can get the shape of the virus particle. And so electron microscopy was used for many years to look at the shapes of different particles. So a couple of considerations about electron microscopy. Most biological materials have very little contrast. You have to stain them. I'm sure you know this. Even looking at materials in a light microscope, you have to put some kind of stain on them to see them. And so with very small particles like viruses, you can't use a dye like crystal violet. You have to use something that's molecular resolution. And so for viruses, we actually do what we call negative staining. We add an electron-dense material, typically uranyl acetate or phosphotungstate, which will scatter electrons. And so these pictures are all negatively stained. So the resolution of this is quite low, 50 to 75 angstroms. So remember, the alpha helix is about 10 angstroms in diameter, so you can't see individual atoms, but you can see the morphology of the virus particle. So remember, when the electrons pass through we have our virus particles on a little grid sitting in the electron microscope, and electrons are used to bombard it, not light as in a light microscope, but we're using electrons. When the electrons pass through the grid, it will expose the film and it will be black, and where the images are white, the electrons are bouncing off. They're not going through to the film. In the old days, they used film. Now they can use digital detectors. So here we have adenovirus on the lower left. The particle itself is white because the electrons aren't going through it, because the particle has absorbed the uranyl acetate. And you can see all around it, it's black because the electrons are going right to the film. And you can see in the particle itself, there are little black areas surrounding the structural units. So there's obviously gaps where the electrons can go through. And the next one is a hepatitis B virus, a spherical virus. Next one over is influenza virus. And you can see, I think, the inside, which is the nucleocapsid. There's not any resolution in there. You can't see RNAs, but you can see on the surface the membrane with little spike glycoproteins in it. So you can get an idea, oh, this virus has something there, and we eventually find out that there's spikes. And then on the right, poliovirus with some broken particles that don't have RNA in it. Anyway, so for many years, this is what we were limited to. Then X-ray crystallography was developed first to study structures of proteins, and then viruses, which were much bigger and required a lot of computational power, which wasn't available until the 70s and the 80s, right? Because all the atoms that you would have, you couldn't possibly order them in a structure without a computer. So what you do here is that you take purified virus and you get it to crystallize. You make actual crystals. So think about salt crystals, right? Or some other element or mineral. Same thing, except you're using big viruses to make these crystals. And you have to figure out how to get them to form crystals. It's not straightforward. Many people have had their lives burned out by trying to make crystals for years and years and years. They give up and they go and do something else with their life. It's a very frustrating endeavor. But once you have a crystal, you then bombard it with X-rays, and then the X-rays are going to bounce off all the atoms in a pattern that will give you those kinds of diffraction patterns over there. You may remember the famous one of DNA that Rosalind Franklin took. It's got a number which I can't remember. She left it on her desk, and supposedly Watson and Crick took it and used it to solve the structure. Well, that's a diffraction pattern where all the dots are resulting from X-rays bouncing off the atoms in the crystal. And then you can use that information along with in the case of a protein, a viral protein, the amino acid sequence to figure out the three-dimensional structure. So I'm skipping a lot of important stuff here, but we don't have time to go into it. The next revolution was cryoelectron microscopy, where you can take virus particles and you simply freeze them in an aqueous solution. And that gives them enough contrast that when you put electrons through them in an electron microscope, you can see the particles. And so what happens is you take your particles, you freeze them very quickly, and then you just put them in the scope and take pictures. And you take a lot of pictures of individual particles. And the idea is every particle is in a slightly different position. It's kind of like a CAT scan, right? Where they put you on a bench and they rotate an X-ray around you to take a three-dimensional, they reconstruct three-dimensional images. It's the same thing here, except the viruses are all moved around independently. You take a few hundred individual pictures, and then you do some mathematics. You do Fourier transformations, figure out how all of these sections are related to each other, you merge them, and then you can do an inverse Fourier transform to get your virus particle. So each of these slices is basically one particle, and then you can put enough together to get the three-dimensional structure. And this is great for it's good for large things. Viruses fit this perfectly. You can do this very quickly because you don't have to make a crystal. You don't have to spend years making a crystal. You just grow virus and you freeze it, and you can do it. And computational advances have made this really, really good over the years. The three people who were essential in developing this are shown here. They won the Nobel Prize in Chemistry in 2017. Each of them developed a different part of this methodology. And a lot of the structures I'm going to show you were based on that. So here's poliovirus, whose structure was solved by X-ray crystallography in 1985. That's shown in the upper left. So the resolution here is 2.9 angstroms. That means we can see things that are 2.9 angstroms apart. And the cryo-EM on the right is 20 angstroms. So you don't have a lot of resolution. And on that right cryo-EM photo, you can see, you don't see individual atoms. You just see the surface of the particle. I mean, you can see features. It's far better than EM. But now cryo-EM is much better. You can get this down to 2 or 3 angstroms or less, all by computational advances, by the way. And you can make images that look very much like this X-ray crystallography image on the upper left, where you can see every atom. You have a file with the three-dimensional coordinate of every atom and use a computer program to build these images. So people have done many, many different virus structures. Here, for example, is Zika virus, which scared the world in 2015 because suddenly it began infecting pregnant mothers and causing microcephaly in the babies. So people worked on this quite a bit. And in the upper left is structure was solved by Cryo-EM very rapidly within 6 months after the virus outbreak began. On the upper left is a space filling model where we represent the atoms as spheres. And you can see a couple of different proteins there making up the virus particle. These are actually glycoproteins embedded in a membrane, and they have an order which we'll talk about in a moment. But on the bottom is the same coordinates, but we're showing a ribbon diagram. So we're not doing space filling. We've thrown out most of the side chains, but we're looking at a ribbon diagram so we can get an approximation of how the chains interact with each other. And you can see on the very top here, some of these ribbons have side chains. Those are sugars, which here's one that you can see nicely in yellow. Glycoproteins that are important for the biological activity. Shortly after SARS-CoV-2 was identified in January of 2020, within a month, the structure of the spike protein anyway was solved by Cryo-EM. So here's the virus particle. It's an envelope virus with a few different proteins embedded in the membrane. And the main one, of course, is the spike protein, which is a trimer of one polypeptide. Of course, most of the vaccines are based on the spike protein. We'll talk about that later. And on the right is the Cryo-EM solved structure of the spike protein. You can see all the different alpha helices and beta strands and so forth that make that up. And we'll talk about what these do in the function of the protein, but very rapidly done because it was clear that it was important. And the technique of Cryo-EM lets you do that. Subsequently, other SARS-CoV-2 protein structures were solved, and a graphics company here in New York City put together this composite image of the particle. So that's the particle on the left there. There's a piece cut out of the membrane, so you can see the nucleocapsid inside, the protein. Nucleocapsid protein, which you may have heard about, it's the basis for the rapid antigen test. It's inside there, attached to the RNA. And then we have the spike protein, whose structure I just showed you, embedded in the membrane as well. And on the right is a close-up of that. So very powerful approaches you can use to solve these virus structures. And you can solve some really, really big virus structures. This is one of the giant viruses. It's called Cafeteria Rohnbergensis virus. And this is a virus that infects this organism. Its name is Cafeteria Rohnbergensis. It's a protozoan that lives in the ocean. In fact, it makes up most of the mass, a great deal of the mass in the ocean. It's the most numerous one in the ocean. And it's called cafeteria because it seems to eat everything. Bacteria, fungi, whatever it can find in the ocean, it eats. So that's why it's called cafeteria. Anyway, this virus infects it. And it's huge. It's 300 nanometer particle, 10 times the size of poliovirus. It has over 15,000 capsid proteins. And this little rotation, so the guy who did this is Xuan Shao, a guy in UTEP. And he's fooling around because this virus infects a protist in the ocean, right? So he's got this thing rolling on the ocean. To do this animation took him 3 million CPU hours, right? He said in the beginning it kept crashing his computer. So you can solve very, very large structures. This is a virus, of course, made up of a pure protein shell. There's no membrane on it. All right, so that's how we solve virus capsid structures. And let's talk now about the different kinds of virus particles that we have. So last time, remember, talked about 7 different kinds of genomes. There are 3 different kinds of virus particles. So 7 genomes, 3 different kinds of virus particles. We have helical virus particles shown there on the upper right. We have icosahedral virus particles there in the middle. And on the bottom, we have complex, which don't have any kind of symmetry. So the first two have some sort of symmetry, helical symmetry or icosahedral symmetry, and I will explain those to you in a moment. The ones on the bottom here have no obvious symmetry, so we call them complex. They include pox viruses, so the agent of smallpox, mpox, which we used to call by another name, right? Mpox is a pox virus. And the giant viruses like Parvovirus and so forth, they all have complex symmetry. All right, so symmetry is important for building many different kinds of virus particles. And this was a concept actually discovered by Watson and Crick. They didn't just look at the structure of DNA. They looked at the structure of virus particles. And they made another contribution to virology that was important. They noticed that if you looked at the electron micrographs, remember we're talking about in the 50s and 60s now, no Xray crystallography. The first Xray structures of viruses weren't until 1985. So in the 50s and 60s, Crick and Watson are looking at EMs of virus particles. And on the top right is tobacco mosaic virus, which is this rod-like virus. And then on the bottom is poliovirus, which is spherical. And they looked at many different viruses and they said, they're either spherical or rod-like. Okay. And so they came up with this idea that because the viral genome is typically small, and I put an exclamation point there because we know it's not small anymore, but back then most of the known genomes were small, it would not be possible to encode a lot of different proteins to make a virus particle. So they thought, well, there must be one or a couple of proteins that are repeated many times to give you the virus particle. So this idea that you would take one or a few proteins and repeat it, this is genetic economy, right? Because you have a small genome, you can't have a lot of proteins in there. We know that many viruses now have big genomes and they encode a lot of structural proteins. But this principle still holds, in fact, not because the genomes are small, but because to make a stable particle, the best thing to do is to take one or a few proteins and repeat them many times. And so they said these two kinds of particles, the rod-like and the spherical ones, are made in distinct ways. So the ones on the top right, that's the rod-like ones, they said these are helical symmetry. We make this by taking proteins and wrapping them around a nucleic acid using helical symmetry. I'll show you that in detail in a moment. And then for the round viruses, they said they're built with icosahedral symmetry. An icosahedron, of course, is a platonic solid. It's one of a number of different platonic solids. And this is one that is well suited, as you will see, to building virus particles. And so there are two rules that we understand now for building virus particles. They explain how virus particles basically self-assemble. You just make the proteins and they assemble themselves. First of all, rule one, every subunit has identical, and I have this in quote, you'll see why in a moment, identical bonding contacts with its neighbors. And it turns out that they're not always identical, but they're almost identical. That's why we have a quote there. And so you have a few proteins repeated over and over. So you repeat the same surfaces of these proteins over and over. That gives you good stability and it gives you a symmetrical arrangement, as you'll see, for both helical and icosahedral symmetry. And then, of course, these contacts are usually non-covalent so that the virus particle can come apart when it gets into a cell, as we've already talked about. But also during assembly, if there's a mistake made, they can be taken apart, kind of like an assembly line for a car, and we'll talk about that later. You know, if you put the door on wrong, or if it's a broken door, you can take it off and put a new one on. Similar idea for these being reversible. And so here's a practical example of the use of symmetry and self-assembly. Many viral capsid proteins, the subunits of the capsid, the individual proteins, if you make them in a cell, they will assemble into a capsid without anything else, without any viral nucleic acid. And we take advantage of this to make vaccines. The hepatitis B vaccine, the human papillomavirus vaccines are made simply by expressing single protein in cells in culture. It makes empty capsids. There's no viral nucleic acid there. We call this virus-like particles, or VLPs. And then that's used to immunize you. So here is the hepatitis B virus vaccine. We make a single polypeptide in culture. That assembles into pentamers, 5 of those polypeptides. So all the information for that assembly is built into the sequence of the protein. And then these pentamers assemble to a virus particle. You can see in the middle here is the original pentamer in the very middle with the different colors that I've shown you on the left. That virus-like particle is then purified, and that's your vaccine. It's just tremendous, and it's based on the idea that these proteins have built within them all of the information to make a capsid. So this is practical, actionable information. Let's talk about helical symmetry first. We've encountered tobacco mosaic virus. The EM is on the lower right, the first virus we discovered. These are rod-like viruses. They're built with helical symmetry. So you know what a DNA helix is, right? This is a similar idea, except it's made of proteins. And so in the simplest case, which is tobacco mosaic virus, there is a single protein, shown here in yellow, which simply interacts with other proteins of the same kind. And it coils around and around and around to make a helix. And in the virus particle, these proteins bind the viral RNA, which is shown here on the upper right. So multiple capsid proteins. So this is the capsid protein subunit. Multiple proteins interact with each other and with the viral RNA. They coil around in a helix, and they make a rod-like virus particle. That's a single protein goes to build this. And of course, now the RNA is protected by that coat protein that's wrapped around and around and around, and only needs to come apart when it gets into a cell. So there are many plant viruses with this kind of helical symmetry. There are also viruses that infect mammals with helical symmetry. Here is one. This is called Sendai virus. It is a paramyxovirus, the same family as, say, measles virus. It has a long nucleocapsid. Again, one protein, this is called the nucleocapsid protein, is binding to the RNA. In this case, it's negative strand RNA, and it coils around and around for the length of the RNA. This is longer than tobacco mosaic virus because the genome is longer. The difference here is that when we have helical symmetry for animal virus particles, they're always enveloped. So here is a paramyxovirus with an envelope. You can see this one's broken and the nucleocapsid is coming out. But animal viruses with helical symmetry are always enveloped, whereas those in plants are not, and not clear why that's the case. Vesicular stomatitis virus, a virus related to rabies virus. This is a virus of cattle. It's also a virus that we use for vectors. We've made many vaccines using VSV. It has helical symmetry. It has a bullet shape, unusual, where one end is sealed off here and the other end is flat. But the nucleocapsid protein, shown right here, has a groove where it binds to the RNA. And here are a number, this 10 or so of these proteins shown binding to an RNA, which you can see in green. And essentially, these proteins coat the entire RNA genome and make a long nucleocapsid, very much like the ones we've already seen for other viruses. And then that nucleocapsid is wrapped in a membrane because this is a virus that infects mammals. So here are some different enveloped RNA viruses with helical capsid. These happen to be negative-stranded RNA viruses. So measles and mumps viruses, paramyxoviruses, are enveloped with a helical nucleocapsid. Again, proteins bound to the RNA. Here's vesicular stomatitis and rabies virus, same helical capsid. And we call it a nucleocapsid, right? Because it's a substructure within the membrane. The tobacco mosaic virus does not have a membrane. It's not a nucleocapsid, it's just a capsid. I shouldn't say just because it's pretty impressive, but it is a capsid in nomenclature, not a nucleocapsid. These are nucleocapsid. Influenza viruses, 8 nucleocapsids within an envelope. And filoviruses, Ebola virus, happens to be much bigger than all these others, but it's simply a nucleocapsid, a protein wrapped around negative strand RNA with a membrane around it. Okay, so the nucleocapsid is the nucleic acid protein assembly packaged within the particle, and it has the name nucleocapsid because it is a substructure. There are also plus RNA viruses with helical capsids. The coronaviruses are great examples. There's the nucleocapsid of a coronavirus, the nucleoprotein plus the RNA, that's the nucleocapsid, and it's a substructure because there's a membrane around it. And these are some electron micrographs of coronaviruses. When they were first discovered many years ago in the 40s, these were the kinds of pictures we would see, and it looked like they had a halo of spikes around them that resembled a solar corona, so they were called coronaviruses for that name. And here on the bottom, one of the first electron micrographs of SARS-CoV-2 taken, very clear that it had a similar morphology, and so likely a coronavirus, confirmed by sequencing of the genome, of course. There are also DNA viruses with helical symmetry. It's not just RNA viruses. We have double-stranded DNA viruses of archaea. We have single-stranded DNA viruses of bacteria. We also have both rod-like and flexible or flexuous RNA viruses of plants. And again, this one has a membrane. This double-stranded DNA virus of archaea has a membrane. So that would be a nucleocapsid in it. But all these others are naked viruses. So these are just capsids. This is what I forgot to bring you. I have these lovely Buckyballs that I have collected over the years. And you can make a helical nucleocapsid with them. And our TA Richard brought me today his childhood collection of Buckyballs. Look at them all. I'm going to have a ball making helical nucleocapsids. Am I embarrassing you that you like to use them? Look at them all. So I always wanted a lot of them. And he said you have no use for these anymore, right? You don't want to play with them. So I'm going to build more of these. But you could build this. And this one is actually made of Buckyballs that many students have given me over the years. They're little magnets, very powerful, and they stick to each other strongly. And you can make a nice helical capsid with them. You can also make icosahedral capsids with them, as I'll show you. But right now, let's take another pause and look at another question here. Which of the following describes virus symmetry and self-assembly? The bonding contacts of subunit are usually covalent. The bonding contacts of subunits are usually non-covalent. Each subunit has different bonding contacts with its neighbors. Self-assembly of virus particles does not occur, or none of the above. What's the answer? Bonding contacts are usually non-covalent is the correct answer. So they're not covalent, right? Otherwise the particle couldn't come apart. Different bonding contacts, no. The whole key of symmetry is you get identical bonding contacts throughout the particle. And self-assembly does not occur. It absolutely does occur. I showed you an example of that for 2 viruses. We get self-assembly. So bonding contacts are usually non-covalent is the right answer. All right, let's talk about icosahedral symmetry. So we've talked about these rod-like viruses, but many viruses are round. So how do you make a round virus from proteins with irregular shapes? I mean, these magnets are kind of misleading because, not all proteins are a little perfect sphere, right? So that's not quite right. But proteins are irregular. And how do we make a spherical virus? Well, again, this is what Watson and Crick figured out. So just an example, that's poliovirus on the left, spherical virus. That's one of the viral capsid proteins. It's not spherical. So how do you build a spherical virus from that? And there are a couple of clues that Watson and Crick went by. First, all the round capsid have precise numbers of proteins, typically multiples of 60. Not always, but typically like 60, 180, 240, and 960. And secondly, the spherical viruses can be tiny or intermediate or really big, but the capsid proteins are always roughly 20 to 60,000 Daltons in molecular weight. And so those 2 pieces of information led Watson and Crick to conclude that spherical viruses are built with icosahedral symmetry. Icosahedral symmetry is based upon an icosahedron, which is a solid with 20 faces, 20 triangular faces, each of which is an equilateral triangle. The assembly of each of those equilateral triangles into an icosahedron gives rise to what we call the axes of symmetry, simply the points around which there are certain numbers of subunits. So for example, there are a dozen 5-fold axes of symmetry in an icosahedron. That is shown in the green. There are 23-fold axes of symmetry, and there are 32-fold axes. And all these mean is that there are 5 copies around each one. So for example, around the 5-fold axis, in this particular structure, there are 5 copies of this triangle. The 3-fold surrounded by 3 copies of the triangle, 1, 2, 3, and the 2-fold is surrounded by 2. An icosahedron allows the formation of a closed shell with the smallest number of identical subunits, and that would be 60. Now, each triangle of this capsid is formed with viral proteins, of course, but viral proteins are not triangular, and so one protein subunit alone is not enough to make a triangular face of an icosahedron. Each face is formed by at least 3 viral protein subunits fitted together to form the face. These 3 subunits can be the same protein, or they can be 3 different proteins. Those 3 proteins together form what we have defined in the beginning as the structural unit. And the structural unit is repeated to form the capsid. So for example, there are 5 structural units around the 5-fold axis of symmetry, forming what looks to be a pentamer, in fact, is a pentamer. All right, so let's see how this works. The simplest icosahedral capsids are made of 60 identical protein subunits. Remember, the subunit is the individual polypeptide chain. And for these viruses, the structural unit, which is what the particle is made from, is a trimer of this one protein. So here's a very simple virus particle. You see it's spherical. It doesn't look like the icosahedron that I had on the previous slide because it's a symmetry rule. It's not necessarily making that kind of a solid. So here's a simple virus particle. It's made of 60 copies of one protein, and you can see the structural unit is outlined in dotted lines there. And there's 1, 2, 3 proteins. The same protein, 3 of them make up the structural unit. And these proteins all interact in the same way. The interactions are identical, head to head, tail to tail, right? So that is where this identical interactions come from. Wherever you go in the particle, the proteins are interacting in a similar way. The protein, by the way, is the comma. So these, again, are spherical. They're not icosahedra. They're typically spherical particles, but icosahedral is the kind of symmetry with which the proteins are arranged. Adeno-associated virus 2, it's a Parvovirus. It is 25 nanometers, pretty small, 60 copies of a single capsid protein. So that's what the particle looks like. And on the left is the single capsid protein, its structure shown here. And these are packed, 60 copies of that is packed here to make the capsid, which contains a single-stranded DNA genome. So this is a very simple animal virus, which as you will see, we use extensively for gene therapy applications. All right, so making a larger virus, we don't use bigger proteins. We just add more of them together. This is an important concept. So, no matter how big the virus, the proteins that make it are all more or less the same size. You just have more of them in a bigger virus. So here is a virus where we have 180 identical protein subunits. Remember, the previous one had 60. This one now has 180. So obviously we've stuffed more into the capsid and it gets bigger. But then there are other consequences of that I want to tell you about. It changes the environment of the proteins. You no longer have identical interactions. For example, you now have pentamers and hexamers. Here's a pentamer, 1, 2, 3, 4, 5 copies of the protein. It's a pentamer. But look here, the purple protein, 1, 2, 3, 4, 5, 6 copies. Hexamer. This is a consequence of adding more proteins in. They can no longer have 5-fold axes. They now have to arrange in pentamers and hexamers. And so you have slightly different interactions in this capsid. The interactions are not all identical as they were in the simple virus. We call them quasi-equivalent because they're similar, but they're not exactly the same environment. So for example, we all have head-to-head and tail-to-tail interactions, but there's pentamers and hexamers, so that's a different environment because here there are 6 proteins and here there are 5, so it's a different environment. So you can make bigger and bigger virus particles simply by adding more proteins. So quasi-equivalence is the name we give for these bigger, anything bigger than 60 is going to have quasi-equivalence. If you have more than 60 subunits, each occupies a quasi-equivalent position, which means that the non-covalent binding properties are similar but not identical, right? Head to head, tail to tail, but you have pentamers and hexamers, which are different environments. Now, one of the key concepts about building icosahedral capsids is the triangulation number T. And this makes it easier for us to understand how adding more subunits to the triangular face increases the size of the particle. So the T number is defined in its simplest fashion as the number of structural units per triangular face of the icosahedron. So here on the bottom is a schematic of an icosahedron. Remember, it's made up of 20 equilateral triangles. Each of those is the triangular face. So the number of structural units per face is the T number. So for example, T equals 1 virus particles, there is 1 structural unit. And for T equals 1 viruses, that is typically made up of 3 of the same protein. You can see the 3 proteins here, gray, red, and blue, form the structural unit, and there is 1 structural unit per triangular face. You can always find triangular faces on the particle by simply joining the 5-fold axes of symmetry. So in this T equals 1 particle here, in purple, you see the 3 5-fold axes of symmetry. We draw lines between them. That's the triangular face. That is the equilateral triangle, the triangular face of the icosahedron. And for T equals 1, you can see there is 1 structural unit within it. Now let's jump to T equals 4 next because it's simpler. And I'll tell you why in a moment. So a T equals 4 triangulation number means there are 4 structural units in the triangular face. 1, 2, 3, 4. And you can see it on the entire capsid below, where we have defined the triangular face by joining 5-fold axes of symmetry. By drawing a line between them, you define a triangular face of the icosahedron, and you can count 1, 2, 3, 4 structural units within it. So that's a T equals 4 virus. Now, the reason I skip T equals 3 is because sometimes the structural unit overlaps from one face to another. So here on the T equals 3 in black, that is the triangular face. T equals 3 has 3 structural units within it, but they overlap from one face to another. So in a T equals 3, 3 structural units form the face, but they occur as 6 half units, right? So here's 1 half unit, a second, a third, a fourth, a fifth, and a 6th half unit. So it's a little tricky. That's why I went to T equals 4 first. But at T equals 3, 3 structural units form the face. Same with T equals 7. 7 structural units form the face, and so on. You can keep increasing the T number to very high numbers. So that's what the T number is, the number of structural units per triangular face. And the fact that the structural units increase as we increase the T number reflects the fact that the virus particle is getting bigger by adding subunits to each triangular face in the form of additional structural units. So you can build these out of Buckyballs too. I didn't know this until a couple of years ago. I went to visit Indiana University. I was in a chemist's office, and he had these on his shelf. I said, how did you build it? So he spent me like an hour teaching me how to build these. And so here's a T equals 1. I made a video of it because this is so cool. Go watch it. It's really, really fun. You probably won't think it's fun, but it's fun. Here's a T equals 1 made up of only pentamers. So you take these magnets, 1, 2, 3, 4, 5, and you can use those to build the particle. 12 of these will build the particle. And I made some of different colors. So you can see 5, 5, 5, 5. That is your simplest virus. Now to build the T equals 3, it's really easy. You get pentamers, and then you make some hexamers out of the Buckyballs, and you assemble them, and you can see the pentamers in red and the hexamers in blue. And that's got 180 subunits. That's the concept of building these virus particles. So let's go through some examples of these. We looked at a T equals 60, a T equals 1 virus, adeno-associated virus. This is a T equals 3 poliovirus. The structural unit is made up of 3 proteins, and there are 3 different proteins. So you could have 3 of the same protein or 3 different proteins. And you don't need to worry about this, but I'm going to tell you in case you run across it. When you have 3 different proteins comprising the structural unit, it's called pseudo T equals 3 symmetry, right? So it's not just T equals 3, it's pseudo T equals 3. Poliovirus, actually 4 different polypeptides. There's one lining the inside of the capsid, which is really an extension of one of these proteins. So VP1, VP2, VP3, individual different polypeptides forming the structural unit. 5 of those form a pentamer, and then 12 pentamers make up the virus. And the virus particle, of course, has 5-fold and 6-fold axes of symmetry, as you would expect for something greater than T equals 1. You can make bigger ones. This is SV40, which we'll be talking about as well. 50 nanometer particles, 72 pentamers of VP1 gives you 360 subunits. And so here is the pentamer. It's VP1, 5 copies of VP1. And then these pentamers are arranged in 5-fold or 6-fold environments. So here we have a VP1 pentamer with 5 neighbors, 1, 2, 3, 4, 5. And here we have a VP1 pentamer with 6 neighbors, 1, 2, 3, 4, 5, 6. So again, as you make anything bigger than T equals 1, you have to have pentamers and hexamers in that. This is one of the viruses that self-assembles, that we'll talk about later. This is a close-up of one of the pentamers, just to show you how the pentamers are locked together, non-covalently, but they're very stable. Let's go back to the previous. The N-terminal extensions of VP1 interact with VP1 of the neighboring pentamer. That's how they stay together. So here, this N-terminal extension is colored in yellow. And you can see how it's wrapped around the N-terminal extension of the neighboring pentamer, which is in blue here. So that makes it very stable. But again, it's non-covalent, so it can come apart to let go of the genome. So we can make even bigger capsids, and then we have other proteins involved that have specialized roles. This is adenovirus, which is 150 nanometers now. So remember, we started with a Parvovirus at about 20 nanometers. Now we're 150. It's a T equals 25 capsids. And this one, so it's an icosahedral shell, as you can see in this electron micrograph. And it has a number of proteins with specialized roles. For example, there is a protein sticking out from each 5-fold axis of symmetry. You can see on this reconstruction, just cryo-EM reconstruction on the bottom here, these green fibers, they're called the fiber protein. And they are how the virus is going to attach to the cell receptor. And so that's one protein with specialized roles. This virus particle is made up of these blue proteins are called hexons. They're made up of a trimer here. And then this is the structure of the fiber. It's made up of a knob, a fibrous shaft, and a penton base that fits into the particle. For example, one of these proteins that has a specialized role is protein 9. That's this one here, the little brown protein that's in between the hexons. So as you make bigger virus particles, these penton-hexon interactions are weaker than, say, penton-penton interactions that you would get in the smaller particles. So as you get bigger, less stable, so you end up having to put other proteins in to compensate. And this is one of them. It simply helps to glue the hexons together. In fact, it's called the cement protein. Here's another example of larger capsid. These are reoviruses or rotaviruses, which we'll talk about as agents of gastroenteritis. They're viruses with double-stranded RNA segments, T equals 13, 70 to 90 nanometers. But the interesting thing here is that there are 2 icosahedral shells. There's an outer shell and an inner shell wrapped around each other. So not just one, but two. There's no envelope here. And we'll see when we talk about uncoating next time that the outer one has to be stripped off in order for uncoating to occur. So it's some kind of solution for stability. But here on the right are the structures of the outer shell, which is made up of trimers of a viral protein with a T equals 13 number. And on the right is the inner shell, which has a T equals 2 number. It's very different T numbers. They're still compatible with being wrapped around each other. The tailed bacteriophages are nice examples of having multiple kinds of symmetry in one particle. So there's the famous tailed bacteriophage. When people see this, they're always fascinated by it because it looks like steampunk, right? But it's really made up of an icosahedral head, which has symmetry, just like other icosahedral capsids do, except it's connected to a tail, which is built with helical symmetry. So two different kinds of symmetry. And there are other proteins here with specialized functions. We have tail fibers, which are involved in helping to attach to the bacterium. And then we have a base plate, where the virus sits down on top of the bacterium and injects the nucleic acid through the base. So here's the base plate in close-up here in panel C. You can see it's made up of a lot of different proteins. That's the tail fiber there. And for many of these viruses, when the virus attaches to the bacterium, the tail fiber actually contracts and shoots the DNA through from the head into the cell. And the DNA in this head is packaged at hundreds of pounds per square inch of pressure. You know, you have a tire in a car, it's 30 or 40 PSI, right? 600 PSI for some of these, and that's how it comes out. So it's packaged in under high pressure. The capsid is strong enough to be able to hold it, and then the base plate sits down on the bacterium, punches a hole in the bacterial wall, and out comes the DNA. So the base plate is really cool. The tip looks just like a spike. In fact, it's called a spike. So there's a phage where the tail has been contracted, and the spike is poking through the bacterial wall. And so the spike protein of this particular phage, whose structure has been solved, as you can see, there it is on the bottom there in red. That's the base plate. There's the tail. So it's in red. And here's the structure of it. It's a trimer made up of 3 copies of the same protein. And you can see the top part is mostly beta sheet, and it gets short. Each strand gets shorter and shorter, so it tapers as it goes down. And then the bottom are just random coils. And there's a molecule of iron down there that coordinates the 3 chains together. So it's a spike. It's just like a spike. And this is what plunges through the bacterial membrane. Presumably it falls off and leaves an opening through which the DNA can come through. So very cool proteins with specialized functions. Another one is the herpes simplex virus capsid. Now remember, this is an envelope virus, and within it is a nucleocapsid composed of a icosahedral shell containing the double-stranded viral DNA. And the icosahedral shell, its structure has been solved. You can see it on the right here. It's big, so as you might expect, this is a T equals 16 capsid. So you will find pentamers and hexamers. Absolutely, you do find them there. And there are lots of proteins with specialized functions. But the one I want to point out to you is this purple one here. It's called the portal. You ever play that game called Portal? It's a cool game, right? I love it. I loved it. Years ago, I was playing it, and my son was watching me. He just grabbed the mouse and finished it in an hour. He said, "Dad, you don't know what you're doing." But anyway, this is the portal of herpes simplex virus. There is 1 portal per capsid. And here we have stained the portal with antibodies. You can see 1 on that particle, 1 on that particle. So the portal is at one of the 5-fold axes of symmetry. And let's see, can we see it on the other? Yes, can't see it on this one. It's not on this one either, I think. Nope, it's not there, but you can see that this is the reconstruction of the portal here, and on the capsid itself, this is an electron micrograph, the portal is right there. This is important because the portal is how you put DNA in the capsid when you're building it, and it's how the DNA gets out as well. So a protein with specialized function. So I have a bunch of these herpes virus keychains. Every year somebody gives me one. I have a lot of them. So, and this is cool because, all right, this is the thing closed. Then you open it up and there's the nucleocapsid, right? And look, they actually put a portal on it right there. One portal, you can tell by the size. And then you can open up the nucleocapsid and you can see the DNA genome inside of it. So a very accurate herpes simplex virus keychain. And I'm not getting any money from this, so I'm just showing it to you because it's cool. All right, finally, capsids can be covered by host membranes, whether they're nucleocapsids or icosahedral capsid. They can be enveloped. The envelope is a lipid bilayer that comes from the host cell, has to come from the host cell because viruses do not encode lipid synthesizing machinery, and it's typically acquired by a process we call budding, where the virus particle forms as membranes. Here is one at the surface of the cell, the plasma membrane. Virus particles start to be assembled, and a little piece of membrane is budding out and eventually pinches off. This can happen at the plasma membrane, it can happen at the nuclear membrane, it can happen at the Golgi. But for any virus, it's a specific process. For coronaviruses, they bud into intracellular compartments. Now, within the envelope, you could have an icosahedral capsid, nucleocapsid, or you could have a helical nucleocapsid. And again, because there's an envelope, whatever is inside is a substructure. So it's a nucleocapsid. And so here is an example. Influenza virus, to illustrate that when you have a membrane, you need to put viral proteins in it so that the virus particles can attach to cells, right? Because if you just had a naked membrane, those particles wouldn't have any specificity for infecting cell. And so the proteins are integral membrane glycoproteins. They have sugars typically attached to them. There's an ectodomain outside of the virus particle. There's the membrane of the virus. And then we have an internal domain typically involved in assembly. And these proteins tend to be oligomers. They're dimers or trimers. And so the influenza virus spikes, which we saw in that EM some time ago, those spikes are made up of trimers of these proteins inserted into the membrane. And these glycoproteins can be perpendicular to the membrane. So here is the influenza virus HA or hemagglutinin trimer, which is perpendicular to the membrane. So that's the purple spike there in the membrane. But other viruses, flaviviruses, which include Zika virus and dengue, the glycoproteins are parallel to the membrane. And so here, this is the structure on the right of Zika virus that I showed you in the beginning. These are the glycoproteins of the virus. The membrane is not showing. It's completely covered by these glycoproteins, but they are laying flat on the membrane, and so that is why they have that kind of look rather than a spiky look, which the influenza virus has. We call the influenza virus envelopes unstructured because the glycoproteins are kind of randomly placed in there, whereas the flavivirus envelopes tend to be structured. They can be because they have interactions between these glycoproteins that give them some icosahedral symmetry, because you could see a five-fold axis of symmetry there. So here are some examples of helical versus icosahedral nucleocapsids. Influenza virus, the nucleocapsid is helical within the membrane. Ebola virus, the nucleocapsid is helical within the membrane. And then on the right, we have icosahedral nucleocapsids. Herpes virus, an envelope nucleocapsid, and toga virus, also an envelope nucleocapsid. And again, in both cases, you have a membrane wrapped around that icosahedral nucleocapsid. So if this herpes virus didn't have a membrane, if in one world herpes viruses were not enveloped, it wouldn't be a nucleocapsid, it would be a capsid. All right, finally, complex virus particles are those where we don't see any symmetry. They include pox viruses and the giant viruses, Pandora virus, pithovirus. They have unusual features. For example, the pithoviruses have this cork-like structure at one end. which comes out to allow the viral genome to exit during infection of a cell. And finally, besides all of these components that we've talked about, structural proteins, accessory proteins like a portal or a cement, glycoproteins, there are many other proteins in virus particles that we'll touch on as we go through this course. There are enzymes in many virus particles. In particular, we're going to spend a lot of time talking about reverse transcriptase and retroviruses. Many viruses have proteases in them to help mature the particle. There are other proteins that are involved in infection. Sometimes there are lipids, glycoproteins. So a virus particle is quite complex. It's not just the proteins that we've talked about today. So next time, we're going to take apart these particles. We're going to see how they attach to host cells, how they get inside, and how the genome comes out, how they become unstable in order to achieve that process.